Hello, everyone. Thanks for joining us for today's webinar. We welcome you all to our webinar today, Insights from Pharmacological Models of Inflammation and Arthritis. My name is Laxananthakrishnan, and I will be your host today. Before we get started with the actual presentation and introduction, I would like to introduce you to some of the technical pieces and logistics of the webinar. A copy of the presentation will be available right after the webinar. If you have any kind of technical difficulties, please raise your hand in the control panel and our technical staff will assist you. To ask a question, please use the Q&A tab in the control panel, and you can send your questions to our panelists. At any point in time during the presentations, if you want to get back to the control panel, you can click on the blue button that's floating up at the top of your screen. Now, with that, I would like to introduce today's speaker, Dr. Dave Grass. Dr. Grass received his doctorate from Columbia University in molecular virology. He did his postdoctoral training at the Sloan Kettering Institute for Cancer, working with Dr. Elizabeth Lazy in molecular immunology. He joined Taconic, formerly DNX Transgenic Sciences, in 1992. As a senior scientist, he was responsible for establishing a transgenic animal model generation service. He also supervised research aimed at the generation of rodent models for inflammatory conditions and human lipoprotein metabolism. Over the past decade, he has authored and co-authored several peer-reviewed publications. He was instrumental in creating the phenotyping platform used for characterizing over more than 200 knockout lines and over 400 compounds. He's currently our Vice President of Business Development. And without further ado, I turn the floor over to Dr. Grass. Dave?
Thanks, Lex. It's a pleasure to be here today to talk to you about our immunology and inflammation program. And today I'm going to focus on some of the induced models that we run. Arthritis, I'll focus on collagen-induced arthritis. For multiple sclerosis, I'll focus on our EAE model. For colitis, I'll show you some validation data on our DSS-induced. And I'll talk a little bit about how we can apply these models to characterize genetically modified mice and compound assessment. I'll also talk about how we try to incorporate genetically modified models into the work that we do. And I'll show you an example using the GFAP luciferase, that stands for glial fibrillary acidic protein, driving the expression of a bioluminescent molecule luciferase, NF-kappa B luciferase, and transgenic mice that express human TNF. And at the end of the presentation, I'd be happy to try to answer any questions that you might have. So the approach that we're taking at Taconic really involves bringing a lot of different things together. It's access to animals, whether that's traditional strains, transgenic animals, or animals that we can create, generate using gene targeting or transgenic technology, and that's what refers to custom model generation solutions. Being able to create cohorts of those animals at a defined health status, and then also being able to characterize all of those animals using our phenotypic characterization program. And we do that to actually do target validation, to validate that a genetic animal might be a good model for disease, and also to do compound profiling with those genetically engineered animals or with traditional models. So I'm going to show how that applies to what we've tried to build with our immunology and inflammation program. And you can see that we have several different disease models that we've tried to build. We have arthritis models, septic shock, multiple sclerosis, pulmonary inflammation, colitis, and dermatitis. In addition, we have models of that assess different aspects of the immune response, such as T-cell proliferation, antibody production, delayed type hypersensitivity, and so on. And in addition, we also have the ability to add different readouts to our disease models. So we can look at cytokines, we can do FACS analysis, we can look at cytokine production in vitro and T and B cell proliferation in vitro. And I'll talk a little bit more about how we can customize these disease models by adding different ex vivo readouts at the end. So first I want to just give you an example. We run 4 different models of inflammatory arthritis. I'm going to focus today on the collagen-induced arthritis model. And one of the unique things about the model and the way we run it is that we not only can run this model on the DBA1 background, but we've also developed it on the C57 Black 6 background. And this shows you some data in terms of how we developed that model. We use different doses of collagen to. I'm going to focus on the panels on the right side of the slide, and you can basically see that with the high dose, we actually are getting an increased clinical score compared with the low dose and with just animals that where we didn't induce arthritis, we just treated them with Freund's adjuvant, but not with collagen. And the data is similar when you look at the swelling. Again, with the high dose of collagen, and we're using chicken collagen for the Black 6 mice, we use a different collagen for DBA. We have significant swelling compared with the lower dose and compared with animals that just got adjuvant. We validated this, and all of the assays that we run, we validate pharmacologically. In this particular case, we use methotrexate, and I think the low dose here was 5 mg per kg, and the high dose was 10 mg per kg of methotrexate. And you can see that when you treat the animals with methotrexate, the animals that have vehicle treated, you can see have the highest clinical score here, have the highest joint swelling here, okay? And the animals with the low dose of methotrexate, there's an increase or improvement in the clinical score. High dose improves it even more, but not to the level of animals that are mock-induced. Same in terms of joint swelling. You see an improvement with the low dose of methotrexate, further improvement with the higher dose, not to the level of animals that have been mock-induced. So this is an example of pharmacological validation, and we can use this assay to characterize different compounds or actually on genetically modified animals. I'm going to switch gears and talk about models of multiple sclerosis, and I'll talk about the MOG peptide-induced EAE. And we do that by treating the animals subcutaneously with MOG peptide and Freund's adjuvant, and then giving on day 1 pertussis toxin IP. And when we do that, you can see that the animals that are, well, first I'll just say the animals that are mock-induced, you can see that we don't have any clinical score. And clinical score is the extent of hind limb paralysis. The animals that are treated with vehicle, where we have induced disease, you can see that they have a significant clinical score. With a low dose of prednisolone, 1 mg per kg, we actually are delaying that clinical score. With the high dose, we actually are alleviating or lowering the clinical score across the time span of the experiment. Now the next slide shows a similar experiment where we've run this EAE model, but in this particular case we're running it on genetically modified animals. So these are animals that express luciferase under the control of the glial fibrillary acidic protein promoter. And that promoter is active in activated astroglial cells. So it's active during times of inflammation in the brain or the spinal cord. And you can see that in the animals that, first let's look at the panel on the upper left-hand side. You can look at the clinical score, and you see that the animals in which we've induced the disease and are treated with vehicle, we have a significant clinical score, and it peaks right around day 17. In this experiment, we used FTY-720 as the treatment, and you can see that that treatment blocks the clinical score. Once we stop the treatment on day 18, you start actually seeing clinical manifestations in that group. When you look on the right-hand side at the animals that we've imaged, and this is one animal per group representative that we're imaging on different days, you can see that in the animals that are treated with vehicle, we have significant light coming from the brain and going down the spinal cord, and that's on day 14. And so the peak actually of this is a surrogate marker for inflammation, and the peak is actually preceding the peak of the clinical score. If we look at the animals that were treated with FTY-720, you can see that it blocks the manifestations of inflammation, or the surrogate marker, the light coming out. And I should mention that what you're looking at here is a false color image where red is the highest density of photons coming from that section of the animal, and blue is the lowest density on the scale. Okay? But you can see that after the treatment, on day 21, just make sure that's day 21, that we have significant inflammation coming down the spinal cord, even though at that point the clinical manifestations are still not that evident. So again, this is giving us additional information that tells us that the inflammation is actually preceding the clinical manifestations, and we can quantify the light coming out with the system that we work with. This slide basically looks at those same animals at the time of the peak clinical score, and we're looking to see if there's inflammatory cells that have infiltrated into the spinal cord. We're looking specifically for CD4 cells and macrophages, and you can see that in this data that in the animals where the disease is evident, we do see CD4 cells and macrophages that have infiltrated into the spinal cord. However, our reference compound, FTY-720, blocks that infiltration of both macrophages as well as CD4 cells. And this data is consistent with what we saw in the imaging on the slide, on the previous slide. The last example that I want to talk about is the DSS-induced colitis model. This model is run by actually administration of 3.5% of DSS in the drinking water for 5 days, after which the animals get regular water. And what we're looking at on this slide is the weight loss that occurs when you treat the animals this way. So animals that are given water all during the experiment, they actually gain some weight. Animals that are treated with 3.5% DSS, you can see that they lose a significant amount of weight. Around 15% of their weight is lost over the course of the experiment, which is 10 days. Now, if we treat the animals with cyclosporin A during this experiment, we have two doses here, 50 mg per kg and 10 mg per kg. And you can see at the high dose, the animals lose very little weight compared with mock-treated animals. The low dose is an improvement over the animals that are treated with vehicle, but not certainly as much as the high dose. So we have a nice dose response in this experiment. Another manifestation, another readout that we look at is the colon length. And as the animals have more inflammation in the intestines, their colon length shrinks. So you can see that animals in which the disease wasn't triggered, they were only treated with water, they have the longest colon length. Animals where we did induce the disease and treated them with vehicle, we see that they have really short colon lengths compared to animals that have the low dose of cyclosporin A, 10 mg per kg, and the high dose, which adds further improvement. So what I'm trying to illustrate with these different examples are that we have a lot of experience in terms of running these types of models. All of the models are pharmacologically validated. And what we try to do is apply these models, these induced models, to characterize both genetically modified animals and to characterize compounds. So for genetically modified animals, we've done a lot of work where we've done target validation work, where we can take a knockout animal and look to see what role that gene might have in these different conditions. One of the advantages and one of the reasons that we try to adapt our models to the Black 6 background is so that we can actually work with these genetically modified animals, most of which are actually made on the Black 6 background. But another utility of being able to characterize genetically modified animals is to be able to characterize models that will be used to test compounds later, so new disease models. And there's a lot of work being done now where specific genes perhaps are being humanized. Those mice have to be characterized to determine whether they still have the same therapeutic window, for example, that the wild-type mice have. And that's work that we have a lot of experience doing. We adapt these assays as well for testing compounds. And we can use wild-type animals, Black 6, as in some of the examples that I showed, as well as I mentioned, genetically modified animals that may be humanized for a particular target. To that end, I should mention that we have a lot of experience with different types of test agents, and especially antibodies, synthetic siRNAs. We've worked with cellular therapeutics as well as small molecules. The humanized mice are particularly important for some of the biologics that need to be tested. In addition, we have a lot of experience working with different formulations, and we have experience with different routes of administration, from oral gavage, subcutaneous injections, IP, intravenous, intramuscular, intradermal, in-diet administration, and we've also used mini pumps. So we have a lot of capabilities in terms of running these types of models. Now, I mentioned before that, and I want to stress the ability that we have to customize each disease model so that we can take the collagen-induced arthritis and add different readouts to it depending on what the mechanism of the potential therapeutic we're testing might be. And the next slide, just there's some points that I want to cover, is that we have a lot of experience looking at cellular infiltrates. I showed you an example where we looked at infiltrates into the spinal cord. We've also looked at cellular infiltrates into the brain, lung, adipose tissue. We do this by FACS analysis. We also have a lot of experience doing single-cell suspensions isolated from the spleen, using various systems, and then we can work with those cells ex vivo to induce different responses. And this gives you more mechanistic information about the either the gene or the compound that you're testing. I've shown you an example of incorporating the glial fibrillary acidic protein luciferase mouse when we ran the EAE model. I'll show you another example just of the NF-kappa B luciferase model, and this follows the regulation of a variety of pro-inflammatory and cytotoxic genes. And just two quick examples here. You can see its activation by bacterial lipopolysaccharide. This is the baseline light coming from the animal. We see a high level of light at 4 hours, less light at 7, and it's going down further at 24. Another example's on the next slide. Activation by TNF-alpha, so we administered TNF-alpha to the mice. Again, this is the baseline level of expression. You see expression at 4 hours, 7 hours, 24. And I should mention that we can look at the full body as well as regions of interest, roughly where the liver is, and quantify the data coming out. One last example I want to show in terms of genetically modified animals that are relevant to studying inflammatory disease is our human TNF-alpha transgenic mouse. This is a mouse that was genetically modified to have deregulated expression of human TNF-alpha. And what that causes is spontaneous arthritis with characteristics similar to human RA. And this is just a picture of the animals at 20 weeks of age. You can see that there's distortion and swelling in the hind paws of these animals compared to the wild type. And these are animals that we can use to do compound testing studies. So I hope that today that I was able to show how we think about building our program and how we've built our program with respect specifically to immunology and inflammation. I've shown you how we run these models on traditional strains, Black 6, on transgenic animals that are modified to express luciferase or to be disease models. And I want to point out that we can create new models, humanized models, with our custom model capabilities, either by gene targeting or by pronuclear injection technologies. We can create cohorts, and I hope that I've shown how we can basically use the capabilities we have to run these models to basically achieve our goal, which is to try to combine all these aspects to create more predictive preclinical data. I'd be happy to take any questions if anyone has any.
Thank you very much, Dave. Again, I would like to remind our attendees that if you'd like to ask a question, you can use the Q&A tab in the control panel, and you can send your questions to our panelists. We have a couple of questions in here. One question here, can you do pain-related readouts for arthritis models? And if so, what type of readouts?
Yeah, we can do pain-related readouts, and we've actually built some of those on. We really study all aspects of pain, so we can do any of those readouts. We do mechanical, thermal, chemical, and neuropathic pain. I guess with the arthritis model, I think we were doing thermal pain on that model as a readout.
Thanks, Dave. Another question in here. What strain of mice is used for MOG-induced EAE model? Is it C57 Black 6?
We actually are using, we actually are running that model on the C57 Black 6 background. There's another model that, and that's thought of, the model that we run most frequently is thought of a chronic EAE model, an acute model. There's a relapsing and remitting model that's run on an SJL background that uses a different peptide, PLP, that we run less frequently, I would say.
There's a follow-up question to that, Dave. The PLP, how do these models, MOG and PLP, differ in pathology?
Say that last word again. I'm sorry.
How do these models, MOG and PLP, differ in pathology?
Okay. Well, the PLP model is relapsing and remitting. So basically, you're having a cycle where where you have an inflammatory condition that resolves and then comes back, and this is, I guess, more similar to what you actually have in the human disease MS. The MOG model that we're running on Black 6 in general is more of an acute model where the condition resolves but it doesn't relapse.
Okay, thanks, Dave. So another question here. Looks like Taconic also offers a TNBS-induced colitis model. How does it differ from the DSS model that you described?
The TNBS model is, well, first of all, the induction is different in that it's induced through the rectal cavity as opposed to DSS, which is induced orally. But the other difference, there are a lot of differences there. The TNBS model doesn't work with a number of different strains, and this is why we've run it less frequently. So it also works in SJL mice, but doesn't work in Black 6 mice very well. We think it's a really good model, and it's something that we could use for compound profiling. It seems to have really mimic the inflammation that you see in colitis, but there's a lot of variability in it, so you need large group, large N's, and most of our clients seem to choose the DSS model because it's a little bit simpler to run.
Thanks, Dave. Another question in here. Is the NF-kappa beta model available off the shelf?
Yes, it is available off the shelf. It is part of, I guess, the Taconic's emerging model program.
I think we have one last question here. Does Taconic get involved or offer any humanized immune system model?
We are starting to do that. We have access to a super immunodeficient mouse, the NOG mouse. That's a SCID mutation on an NOD background with an IL-2 receptor gamma targeted mutation. It's T, B, and NK cell deficient, and so it's got a very compromised immune system. And you can implant cord stem cells from cord blood, CD34 cells, and get some aspects of the human system on the mouse. You could also implant PBMCs there as well. Currently, we don't offer that, but we hope to be doing that shortly. We have a collaboration with another company where we're working to develop those capabilities.
Thanks, Dave. We have another question here. Can you talk about the differences of CIA, AIA, and KRN models of arthritis?
Sure. The CIA model starts by immunizing with collagen. The antibody-induced arthritis model, you're bypassing that initial immune response. So it's similar in that sense, but it's bypassing that response. And KRN is you're basically taking serum from which has antibodies from a transgenic mouse that and that causes the arthritis. So those are the 3 different models. Now, one thing I should mention about the KRN is that it is only available to people who have licenses to that particular mouse. You have to have a license to work with KRN. There's a restriction there. So we can run that model for people who have that intellectual property, but we can't run it for everybody.
That looks like all the questions there today. I want to thank everybody for taking the time to listen to our webinar today, and I hope you can join us for our future webinars. Again, I would like to take the time to thank you for attending today's webinar. Thank you, everybody, and have a good day.